The Biology of Belief by Bruce Lipton, Ph.D. Chapter 3, The Magical Membrane. Now that we've looked at the protein assembly machinery of the cell, debunked the notion that the nucleus is the brain of the cellular operation, and recognized the crucial role the environment plays in the operation of the cell, we're on to the good stuff, the stuff that can make sense of your life and give you insight into ways of changing it. This chapter puts forth my nominee for the true brain that controls cellular life, the membrane. I believe that when you understand how the chemical and physical structure of the cell's membrane works, you'll start calling it, as I do, the magical membrane, or alternatively, capitalizing on the fact that part of the word is a homophone for brain. I refer to it in my lectures as the magical membrane. And when you couple your understanding of the magical membrane with an understanding of the exciting world of quantum physics that I'll present in the next chapter, you will also understand how wrong the tabloids were in 1953. The true secret of life does not lie in the famed double helix. The true secret of life lies in understanding the elegantly simple biological mechanisms of the magical membrane. The mechanisms by which your body translates environmental signals into behavior. When I started studying cell biology in the 1960s, the idea that the membrane was the cell's brain would have been considered laughable. And I have to concede that the membrane in those days was a sorry-looking Mensa candidate. The membrane seemed to be just a simple, semi-permeable, three-layered skin that held the contents of the cytoplasm together. Think saran wrap with holes. One reason scientists misjudge the membrane is that it is so thin. Membranes are only 7/millionths of a millimeter thick. In fact, they are so thin that they can only be seen with an electron microscope, which was developed after the Second World War. So it wasn't until the 1950s that biologists could even confirm that cell membranes exist. Up until that time, many biologists thought the cell's cytoplasm held together because it had a jello-like consistency. With the aid of microscopes, biologists learned that all living cells have membranes and that all cell membranes share the same basic three-layered structure. However, the simplicity of that structure belies its functional complexity. Cell biologists gained insight into the amazing abilities of the cell membrane by studying the most primitive organisms on this planet, the prokaryotes. Prokaryotes, which include bacteria and other microbes, consist only of a cell membrane that envelops a droplet of soupy cytoplasm. Though prokaryotes represent life in its most primitive form, they have purpose. A bacterium does not bounce around in its world like a ball in a pinball machine. A bacterium carries out the basic physiological processes of life like more complicated cells. A bacterium eats, digests, breathes, excretes waste matter, and even exhibits neurological processing. They can sense where there is food and propel themselves to that spot. Similarly, they can recognize toxins and predators and purposely employ escape maneuvers to save their lives. In other words, prokaryotes display intelligence. So, what structure in the prokaryotic cell provides its intelligence? The prokaryote cytoplasm has no evident organelles, such as the nucleus and mitochondria, that are found in more advanced eukaryote cells. The only organized cellular structure that can be considered a candidate for the prokaryote's brain is its cell membrane. Bread, butter, olives, and pimentos. As I came across the realization that membranes were characteristic of all intelligent life, I focused my attention on understanding their structure and function. I came up with a gastronomic treat, to illustrate the basic structure of the membrane. The treat consists of a bread and butter sandwich. To further refine the analogy, I added olives. Actually, my instructive sandwich features 2 kinds of olives, one stuffed with pimentos and the other's pimento-free. Gourmands don't groan. When I've left this sandwich out of my lectures, repeat members of the audience have asked me where it went. Here's an easy experiment to show you how the sandwich membrane works. Make a bread and butter sandwich, at the moment free of olives. This sandwich represents a section of the cell membrane. Now, pour a teaspoon of colored dye on top of that sandwich, as shown in this picture. As illustrated below, the dye seeps through the bread but stops when it gets to the butter because the oily substance in the middle of the sandwich provides an effective barrier. Seen here. Now let's make a bread and butter sandwich with stuffed and unstuffed olives, such as this. Now when we add the dye to the bread, slice the sandwich, we see a different result. When the dye hits a pimento stuffed olive, it stops as surely as it stopped when it hit the butter. But when the dye reaches the olive without a pimento, the pitted olive provides a channel through which the dye can flow freely across the middle of the sandwich, then through the bread to the plate, as seen here. The plate in this analogy represents the cell's cytoplasm. By passing through the pimento-free olive, the dye penetrates the buttery layer to reach the other side of the membrane sandwich. The dye has successfully navigated the formidable fatty membrane barrier. It is important for the cell to allow molecules to break through the barrier because, in my sandwich analogy, the dye is life-sustaining food. If the membrane were simply a bread and butter sandwich, it would provide a fortress-like barrier that keeps out the cacophony of innumerable molecular and radiant energy signals that make up a cell's environment. But the cell would die if the membrane were such a fortress because it would get no nutrients. When you add the pimento-free olives, which allow information and food into the cell, the membrane becomes a vital and ingenious mechanism, enabling selected nutrients to penetrate the interior of the cell, just as the teaspoonful of dye made its way to the plate. In real-life cellular biology, the bread and butter portion of the sandwich represents the membrane's phospholipids, one of the 2 major chemical components of the membrane. The other major chemical components are the olive proteins, which we'll get to below. I call phospholipids schizophrenic because they are composed of both polar and nonpolar molecules. The fact that phospholipids contain both polar and nonpolar molecules may not sound like a recipe for schizophrenia to you, but I assure you it is. All the molecules in our universe can be divided into nonpolar and polar categories based on the type of chemical bonds that hold their atoms together. The bonds among polar molecules have positive and/or negative charges, hence their polarity. These molecules' positive and negative charges cause them to behave like magnets, attracting or repelling other charged molecules. Polar molecules include water and things that dissolve in water. Nonpolar molecules include oil and substances that dissolve in oil. There are no positive or negative charges among their atoms. Remember the adage, water and oil don't mix. Neither do oily nonpolar and watery polar molecules. To visualize the lack of interaction between polar and nonpolar molecules, think of your bottle of Italian salad dressing. You do your best to get vinegar and oil to bond by shaking the bottle, but when you settle the bottle down, they separate. That's because molecules, like people, prefer environments that offer them stability. For their stability, polar or vinegar molecules seek out watery polar environments, and nonpolar or olive oil molecules seek out nonpolar environments. Phospholipid molecules comprise of both polar and nonpolar lipid regions, have a difficult time in seeking stability. The phosphate portion of the molecule is motivated to seek water, while its lipid portion abhors water and seeks stability by dissolving in oil. Getting back to our sandwich, the membrane's phospholipids are shaped like lollipops with an extra stick. See the illustration above. The round part of the lollipop has polar charges among its atoms. It corresponds to the bread of our sandwich. The molecule's two stick-like portions are nonpolar. They correspond to the butter part of our sandwich. Because the butter portion of the membrane is nonpolar, it does not let positively or negatively charged atoms or molecules pass through it. In effect, this lipid core is an electrical insulator, a terrific trait for a membrane designed to keep the cell from being overwhelmed by every molecule in its environment. But the cell could not survive if the membrane were the equivalent of a simple bread and butter sandwich. Most of the cell's nutrients consist of charged polar molecules. It would not be able to get past the formidable nonpolar lipid barrier. Neither could the cell excrete its polarized waste products. Going back to the diagram, I'll read the description of it. Electron micrographs showing the cell membrane at the surface of a human cell. The light-dark layering of the cell membrane is due to the ordering of the barrier's phospholipid molecules. See the inset. The lighter center of the membrane, the equivalent of the butter in our sandwich, represents the hydrophobic zone formed by the legs of the phospholipids. The dark layers above and below the central lipid zone, the equivalent of the bread slices, represent the molecule's water-loving phosphate heads. There, the layers. Integral membrane proteins. The olives in our sandwich are the truly ingenious part of the membrane. These proteins allow nutrients, waste materials, as well as other forms of information to be transported across the membrane. The protein, olives, allow not just any old molecules to get into the cell, but only those molecules necessary for the smooth functioning of the cytoplasm. In my sandwich, the olives represent integral membrane proteins, or IMPs. These proteins embed themselves into the butter layer of the membrane, just as I have embedded olives in the illustration. How do IMPs embed themselves into the butter? Remember that proteins are composed of a linear backbone assembled from linked amino acids. Of the 20 different amino acids, some are water-loving, polar molecules, and some are hydrophobic, nonpolar molecules. When a region of the protein's backbone is made up of linked hydrophobic amino acids, this segment of the protein seeks stability by finding an olive oil-loving environment like the membrane's lipid core. See the arrow below. That's how hydrophobic parts of the protein integrate themselves into the middle layer of the membrane. Because some regions of the protein's backbone are made up of polar amino acids and other regions are nonpolar, the protein strand will weave itself in and out of the bread and butter sandwich. There are a lot of IMPs with lots of different names, but they can be subdivided into two functional classes: receptor proteins and effector proteins. Receptor IMPs are the cell's sense organs, the equivalent to our eyes, ears, nose, taste buds, etc. Receptors function as molecular nanoantennas, attuned to respond to specific environmental signals. Some receptors extend inward from the membrane surface to monitor the internal milieu of the cell. Other receptor proteins extend from the cell's outer surface, monitoring external signals. Like other proteins, which we discussed earlier, Receptors have an inactive and an active shape and shift back and forth between those conformations as their electrical charges are altered. When a receptor protein binds with an environmental signal, the resulting alteration to the protein's electrical charges causes the backbone to change shape, and the protein adopts an active conformation. Cells possess a uniquely tuned receptor protein for every environmental signal that needs to be read. Some receptors respond to physical signals. One example is an estrogen receptor, which is specifically designed to complement the shape and charge distribution of an estrogen molecule. When estrogen is in its receptor's neighborhood, the estrogen receptor locks onto it, as surely as a magnet picks up paper clips. Once the estrogen receptor and the estrogen molecule bind in a perfect lock and key fit, the receptor's electromagnetic charge changes, and the protein shifts into its active conformation. Similarly, histamine receptors complement the shape of histamine molecules, and insulin receptors complement the shape of insulin molecules. Receptor antennas can also read vibrational energy fields such as light, sound, and radio frequencies. The antennas on these energy receptors vibrate like tuning forks. If the energy vibration in the environment resonates with the receptor's antenna, it will alter the protein's charge, causing the receptor to change shape. I'll cover this more completely in the next chapter, but I'd like to point out now that because receptors can read energy fields, the notion that only physical molecules can impact cell physiology is outmoded. Biological behavior can be controlled by invisible forces, including thought, as well as it can be controlled by physical molecules like penicillin, a fact that provides the scientific underpinning for pharmacological-free energy medicine. Receptor proteins are remarkable, but on their own, they do not impact the behavior of the cell. While the receptor provides an awareness of environmental signals, the cell still has to engage in an appropriate life-sustaining response. That is the venue of the effector proteins. Taken together, the receptor effector proteins are a stimulus response mechanism comparable to the reflex action that doctors typically test during physical examinations. When a doctor taps your knee with a mallet, a sensory nerve picks up the signal. The sensory nerve immediately passes on the information to the motor nerve that causes the leg to kick. The membrane's receptors are the equivalent to sensory nerves, and the effector proteins are the equivalent of action-generating motor nerves. Together, the receptor-effector complex acts as a switch, translating environmental signals into cellular behavior. It is only in recent years that scientists have realized the importance of the membrane's IMPs. They are in fact so important that studying the way IMPs work has become a field its own called signal transduction. Signal transduction scientists are busily classifying hundreds of complex information pathways that lie between the membrane's reception of environmental signals and the activation of the cell's behavior proteins. The study of signal transduction is catapulting the membrane to center stage, just as the field of epigenetics is highlighting the role of the chromosomes proteins. There are different kinds of behavior controlling effector proteins because there are lots of jobs that need to be done for the smooth functioning of the cell. Transport proteins, for example, including an extensive family of channel proteins that shuttle molecules and information from one side of the membrane barrier to the other. Which brings us back to the pimentos in our bread, butter, and olive sandwich. Many channel proteins are shaped like a tinely wound sphere, resembling the pimento-stuffed olives in our pictures. When the electrical charge on the protein is altered, the protein changes shape, a change that creates an open channel running through the protein's core. Channel proteins are actually two olives in one, depending on their electrical charge. In the active mode, their structure resembles a pimento-free olive with an open gate. In their inactive mode, The protein shape resembles a pimento stuffed olive that stays closed to the world outside the cell. The activity of one specific channel type, sodium-potassium ATPase, merits special attention. Every cell has thousands of these channels built into the membrane. Collectively, their activity uses almost half of your body's energy every day. This channel opens and closes so frequently that it resembles a revolving door in a department store on the day of a big sale. Every time this channel revolves, it shuttles 3 positive charged sodium atoms out of the cytoplasm and simultaneously emits 2 positive charged potassium atoms into the cytoplasm from the environment. Sodium-potassium ATPase not only uses up a lot of energy, it also creates energy as surely as store-bought batteries provide energy for Game Boys, at least until your kids will wear them out. Actually, the energy-producing activity of sodium-potassium ATPase is a lot better than the batteries your kids wear out because it turns the cells into a constantly recharging biological battery. Here's how sodium-potassium ATPase manages that trick. Every revolution of sodium-potassium ATPase throws more positive charges out than it lets into the cell, and there are thousands of these proteins in each cell. As these proteins go through hundreds of cycles per second, the inside of the cell becomes negatively charged while the outside of the cell becomes positively charged. The negative charge below the membrane is referred to as the membrane potential. Of course, the lipid, i.e. butter portion of the membrane, does not let charged atoms cross the barrier, so the internal charge stays negative. The positive charge outside the cell and the negative charge inside make the cell essentially a self-charging battery, whose energy is used to empower biological processes. Another variety of effector protein, cytoskeletal proteins, regulate the shape and motility of cells. A third variety, called enzymes, break down or synthesize molecules, which is why enzymes are sold in your local health food store as a digestive aid. When activated, all forms of effector proteins, including channels, cytoskeletal, and enzyme proteins, or their byproducts, can also serve as signals that activate genes. These IMPs, or their byproducts, provide signals that control the binding of the chromosomes regulatory proteins that form a sleeve around the DNA. In contrast to conventional wisdom, genes do not control their own activity. Instead, it is the membrane's effector proteins operating in response to the environmental signals picked up by the membrane's receptors, which control the reading of genes so that worn-out proteins can be replaced or new proteins can be created. How the brain works. Once I understood how IMPs worked, I had to conclude that the cell's operations are primarily molded by its interactions with the environment, not by its genetic code. There is no doubt that the DNA blueprints stored in the nucleus are remarkable molecules, which have been accumulated over 3 billion years of evolution. But as remarkable as these DNA blueprints are, they do not control the operations of the cell. Logically, genes cannot pre-program a cell or organism's life because cell survival depends on the ability to dynamically adjust to an ever-changing environment. The membrane's function of interacting intelligently with the environment to produce behavior makes it the true brain of the cell. Let's put the membrane to the same brain test to which we put the nucleus. When you destroy its membrane, the cell dies, just as you would if your brain were removed. Even if you leave the membrane intact, destroying only its receptor proteins, which can easily be done with digestive enzymes in the lab, the cell becomes brain dead. It is comatose because it no longer receives the environmental signals necessary for the operation of the cell. The cell also becomes comatose when the membrane's receptor proteins are left intact and its effector proteins are immobilized. To exhibit intelligent behavior, cells need a functioning membrane with both receptor, or the awareness, and effector, the action. These protein complexes are the fundamental units of cellular intelligence. Technically, they may be referred to as units of perception. The definition of perception is awareness of the elements of environment through physical sensation. The first part of the definition describes the function of receptors IMPs. The second part of the definition, the creation of a physical sensation, sums up the role of the effector proteins. By examining these basic units of perception, we have engaged in an ultimate reductionist exercise, taking the cell down to its fundamental nuts and bolts. In this regard, it is important to note that at any given time, there are up to hundreds of thousands of such switches in a cell membrane. Consequently, the behavior of a cell cannot be determined by examining an individual switch. The behavior of a cell can only be understood by considering the activities of all the switches at any given time. That is a holistic, not reductionistic approach, which I'll elaborate on in the next chapter. At the cellular level, the story of evolution is largely the story of maximizing the number of basic units of intelligence, the membrane's receptor effector proteins. Cells become smarter by utilizing their outer membrane surface more efficiently and by expanding the surface area of their membranes so that more IMPs can be packed in. In primitive prokaryote organisms, the IMPs carry out all of its fundamental physiological functions, including digestion, respiration, and excretion. Later in evolution, portions of the membrane that carry out these physiological functions go inside, forming a membranous organelles that are characteristic of eukaryotic cytoplasm. That leaves more membrane surface area available to increase the number of perception IMPs. In addition, the eukaryote is thousands of times bigger than the prokaryote, resulting in a tremendous increase in membrane surface area, i.e., a whole lot more room for IMPs. The end result is more awareness, which translates to greater survivability. Through evolution, the cell membrane surface expanded, but there was a physical limitation to that expansion. There is a point at which the thin cell membrane was not strong enough to contain a larger mass of cytoplasm. Think what happens when you fill a balloon with water. As long as the balloon is not overfilled, it is strong and can be passed around. However, if you exceed the balloon's water capacity, the balloon ruptures easily, spilling its contents, just as a membrane with too much cytoplasm would inevitably rupture. When the cell membrane reached the critical size, the evolution of the individual cell reached its limit. That's why, for the first 3 billion years of evolution, single cells were the only organisms on this planet. That situation changed only when cells came up with another way to increase awareness. In order to get smarter, cells started banding together with other cells to form multicellular communities, through which they could share their awareness, as I explained in Chapter 1. To review, the functions required for a single cell to stay alive are the same functions required by a community of cells to stay alive. But cells started to specialize when they formed multicellular organisms. In multicellular communities, there is a division of labor. The division of labor is evident in the tissues and organs that carry out specialized functions. For example, in the single cell, respiration is carried out by the mitochondria. In the multicellular organism, the mitochondrial equivalent for respiration are the billions of specialized cells that form the lungs. Here's another example. In the single cell, movement is created by the interaction of cytoplasmic proteins called actin and myosin. In a multicellular organisms, communities of specialized muscle cells handle the job of generating motility, each endowed with massive quantities of actin and myosin proteins. I repeat this information from the first chapter because I want to emphasize that while it is the job of the membrane in a single cell to be aware of the environment and set in motion an appropriate response to the environment, in our bodies, those functions have been taken over by a specialized group of cells we call the nervous system. Though we've come a long way from unicellular organisms, I believe, as I mentioned before, that studying single cells is an instructive way of studying complicated multicellular organisms. Even the most complex human organ, the brain, will reveal its secrets more readily when we know as much as we can about the membrane, the cell's equivalent of a brain. The secret of life. As you have learned in this chapter, scientists have recently made great progress towards unraveling the complexity of the simple-looking membrane. But even 20 years ago, the rough outlines of the membrane's functions were known. In fact, it was 20 years ago when I first realized how understanding the workings of the membrane could be life-changing. My eureka moment resembled the dynamics of supersaturated solutions in chemistry. These solutions, which look like plain water, are fully saturated with a dissolved substance. They are so saturated that just one more drop of the solution causes a dramatic reaction in which all of the dissolved materials instantly coalesce into a giant crystal. In 1985, I was living in a rented house on the spice-drenched Caribbean island of Grenada, teaching at yet another offshore medical school. It was 2 a.m., and I was up revisiting years of notes on the biology, chemistry, and physics of the cell membrane. At the time, I was reviewing the mechanics of the membrane, trying to get a grasp of how it worked as an information processing system. Then that is when I experienced a moment of insight that transformed me, not into a crystal, but into a membrane-centered biologist who no longer had any excuses for messing up his life. At the early morning hour, I was redefining my understanding of the structural organization of the membrane, staring first with the lollipop-like phospholipid molecules and noting that they arranged in the membrane like regimented soldiers on parade in perfect alignment. By definition, a structure whose molecules are arranged in regular, repeated pattern is defined as a crystal. There are two fundamental types of crystals. The crystals that most people are familiar with are hard and resilient minerals like diamonds, rubies, and even salt. The second kind of crystal has a more fluid structure, even though its molecules maintain an organized pattern. Familiar examples of liquid crystals include digital watch faces and laptop computer screens. To better understand the nature of a liquid crystal, let's go back to the soldiers on parade. When the marching soldiers turn a corner, they maintain their regimented structure, even though they're moving individually. They're behaving like a flowing liquid, yet they do not lose their crystalline organization. The phospholipid molecules of the membrane behave in a similar fashion. Their fluid crystalline organization allows the membrane to dynamically alter its shape while maintaining its integrity, a necessary property for a supple membrane barrier. So, in defining this character of the membrane, I wrote, "The membrane is a liquid crystal." Then I started thinking about the fact that a membrane with just phospholipids would be simply a bread and butter sandwich without the olives. In the experiment described earlier, the colored dye could not get through the lipid butter layer. The bread and butter sandwich is a non-conductor. However, when you include the IMP, olives, you realize that the membrane conducts something across while keeping other things out. So I continued writing my description of the membrane by adding, "The membrane is a semiconductor." Lastly, I wanted to include in my description the two most common kinds of IMPs. These are the receptors and a class of effectors called channels because they provide the all-important means for the cell to let in nutrients and let out waste matter. I was about to write that the membrane contains receptors and channels when I realized that a synonym for receptor is the word gate. So instead, I completed my description by writing, "The membrane contains gates and channels." I sat back and reviewed my new description of the membrane. The membrane is a liquid crystal semiconductor with gates and channels. What hit me right away was the fact that I had recently heard or read the very same phrase, though at the moment I didn't know where I had come across it. One thing was for sure, it was not in the context of biological science. As I leaned back in my chair, my attention was drawn to the corner of my desk where my new smiley face Macintosh, my first computer, was parked. Lying beside the computer was a copy of a bright red book called Understanding Your Microprocessor. I had just bought this non-technical paperback guide to how computers work from a Radio Shack outlet. I grabbed the book and found in the introduction a definition of a computer chip that read, "A chip is a crystal semiconductor with gates and channels." For the first second or two, I was struck by the fact that the chip and cell membrane shared the same technical definition. I spent several more intense seconds comparing and contrasting biomembranes with silicon semiconductors. I was momentarily stunned when I realized that the identical nature of their definitions was not a coincidence. The cell membrane was indeed a structural and functional equivalent, or homologue, of a silicon chip. 12 years later, an Australian research consortium headed by B.A. Cornell published an article in Nature that confirmed my hypothesis that the cell membrane is a homologue of a computer chip. The researchers isolated a cell membrane and attached a piece of gold foil under it. They then flooded the space between the gold foil and the attached membrane with a special electrolyte solution. When the membrane's receptors were stimulated by a complementary signal, the channels opened and allowed the electrolyte solution across the membrane. The foil served as a transducer, an electrical pickup device, which converted the electrical activity of the channel into a digital readout on a screen. This device, created for the study, demonstrates that the cell membrane not only looks like a chip, but also functions like one. Cornell and Associates successfully turned a biological cell membrane into a digital readout computer chip. So what's the big deal, you ask? The fact that the cell membrane and a computer chip are homologues means that it is both appropriate and instructive to better fathom the workings of a cell by comparing it to a personal computer. The first big deal insight that comes from such an exercise is that computers and cells are programmable. The second corollary insight is that the programming lies outside the computer or cell. Biological behavior and gene activity are dynamically linked to information from the environment, which is downloaded into the cell. As I conjured up a biocomputer, I realized that the nucleus is simply a memory disk, a hard drive containing the DNA programs that encode the production of proteins. Let's call it the double helix memory disk. In your home computer, you can insert such a memory disk containing a large number of specialized programs like word processing, graphics, and spreadsheets. After you download those programs into active memory, you can remove the disk from the computer without interfering with the program that is running. When you remove the double helix memory disk by removing the nucleus, the work of the cellular protein machine goes on because the information that created the protein machine has already been downloaded. Enucleated cells get into trouble only when they need the gene programs in the ejected double helix memory disk to replace the old proteins or make different proteins. I had been trained as a nucleus-centered biologist as surely as Copernicus had been trained as an Earth-centered astronomer. So it was with a jolt that I realized that the gene-containing nucleus does not program the cell. Data is entered into the cell or computer via the membrane's receptors, which represents the cell's keyboard. Receptors trigger the membrane's effector proteins, which act as the cell or the computer's central processing unit, or CPU. The CPU effector proteins convert environmental information into the behavioral language of biology. I realized in those early morning hours that even though biological thought is still preoccupied with genetic determinism, leading-edge cell research, which continues to unfold the mystery of the magical membrane in ever more complex detail, tells a far different story. At the moment of transformation, I was frustrated because there was no one with whom I could share my excitement. I was alone out in the country. My house didn't have a telephone. Because I was teaching at a medical school, I realized that there would undoubtedly be some students studying in the library. I hastily threw some clothes on and raced off to the school to tell someone, anyone, of this exciting new insight. Running into the library, out of breath, wild-eyed, with my hair flying in all directions, I was the epitome of the absent-minded professor. I spotted one of my first-year medical students and ran up to him proclaiming, "You have to hear this. This is great shit." I remember in the back of my mind how he pulled away from me, almost in fear of this raving, mad scientist who wildly broke the silence of the sleepy library. I immediately began to spew forth my new understanding of the cell, using the complex polysyllabic jargon of a conventional cell biologist. When I finished my explanation and was silent, I was waiting to hear his congratulations or at least bravo, but nothing was forthcoming. He was now wide-eyed himself. All he could say was, "Are you okay, Dr. Lipton?" I was crushed. The student had not understood a word I had said. In hindsight, I realized that as a first semester medical student, he did not have enough scientific background or vocabulary to make any sense out of my apparent rantings. However, the wind was knocked out of my sails. I held the key to the secret of life, and there was no one who could understand me. I confess, I didn't have much better luck with most of my colleagues who had been schooled in polysyllabic jargon. So much for the magical membrane. Over the years, I gradually honed my presentation about the magical membrane and continued to refine it so that first-year medical students and lay people could understand it. I've also continued to update it with the latest research. In so doing, I found much more receptive audiences among a wider range of people. I have also found audiences receptive to the spiritual implications of my eureka moment. Shifting to membrane-centered biology was exciting for me, but it wouldn't have been enough to send me screaming to the library. That Caribbean moment not only transformed me into a membrane-centered biologist, it also transformed me from an agnostic scientist to a card-carrying mystic who believes that eternal life transcends the body. I'll get to the spiritual part of the story in the epilogue. For the moment, let me reiterate the lessons of the magical membrane, which put the control of our lives not in the genetic roll of the dice at conception, but in our own hands. We are the drivers of our own biology, just as I am the driver of this word processing program. We have the ability to edit the data we enter into our biocomputers, just as surely as I can choose the words I type. When we understand how IMPs control biology, we become masters of our fate, not victims of our genes.